Sing you of sinew when you sing a new tune of when you grew anew. All right, well, I've already talked about Avicenna a little bit, I think on the first day, but put just a bit of history back in. We haven't done much history in a few days. Here's a quote from his text that he wrote back in the 11th century. The power of locomotion is that which contracts and relaxes the muscles whereby the members and joints are moved, extended or flexed. This power reaches the limbs by way of the nerves, and there are as many forms of power as there are of movement. Each muscle has its own peculiar purpose, and it obeys the degree of composite sense. All right, well, that is a pretty succinct description of what the class is going to be about today. The first person to go, the first European to go into very exquisite detail in recording the earliest very thorough recording of the human musculature was done by Andreas Vesalius, who lived in the 16th century. He was a Flemish physician who worked in Italy and really established Italy as a hub for anatomical study during the Renaissance. All right, so that's all the history I'm going to talk about. Here's a picture of the neuromuscular junction, and much of the class today is going to be focused on just this structure right here. So in this fuchsia color, we have muscle cells, individual skeletal muscle cells, and in purple, what looks like an alien grasping onto it, that is simply your neuron, a muscle neuron. And this area right here, I should get a pointer, that we see right here, this is the motor end plate, the neuromuscular junction, where the nerve comes in direct contact with the muscle cell and forms a type of synapse. We call it the neuromuscular junction. All right, so as I said in the first day of class, muscle tissue is broken up into three types: skeletal, smooth, and cardiac muscle. And they all have similarities with one another, but distinct differences. So, of course, they're all contractile, excitable tissue. However, cardiac muscle as well as smooth muscle are both involuntary muscle. You can consciously control, some people can consciously control the rate of beating of your heart, but you can't just say, okay, heart, beat now, stop now. It's involuntary. Both cardiac and smooth muscle are mononucleate, meaning they have a single nuclei containing the genetic material. The difference between the two is that the cardiac muscle is striated and smooth muscle is not. And then there's skeletal muscle, which is voluntary, is polynucleate, and we'll talk about that, the reasoning for that in a bit. And it is striated like cardiac muscle. So it is the specific biomolecular mechanisms of contractility that give rise to this striation, which we're going to talk about here for the rest of class. A skeletal muscle itself is made up of all four tissue types. So we have, of course, the skeletal muscle, the muscle tissue, the contractile muscle tissue. We have nervous tissue, which is the job of which is to excite and stimulate the muscle tissue. Then there are connective tissues, which are separating, supporting, and guiding the muscle tissue, as well as a place for epithelium in the blood vessels that are absolutely essential to muscle function. All right, so let's talk about the connective tissue first. There are a lot of parallels between, despite their separate embryological origins, there are a lot of parallels between muscle tissue and nerve tissue, and we'll see those in the next lecture on Monday when we talk about the CNS. And one of them is in terms of the connective tissue. So there are 3 layers of connective tissue in muscle. The outer sleeve of connective tissue that goes around an entire muscle is called epimysium. Epimysium. So it's primarily made of collagen. It's a tough, fibrous shell around a muscle, and it is in close contact with the deep fascia of the body wall. It helps to separate a muscle from surrounding tissue, helps a muscle to move in relationship to the surrounding tissue if it needs to when it contracts. So here we have some muscle taken from this guy's forearm, and we see that it is surrounded by this sheath of epimysium. Now, within a single muscle, we are going to pull out a single muscle, skeletal muscle. We're going to pull out a muscle fascicle. So that's one of these bundles here. If we, in that top panel, we see a skeletal muscle, and then they're indicating each of these little bundles. We've pulled one of the bundles out. That's a muscle fascicle. A muscle fascicle is surrounded by a different type of connective tissue called paramecium. And the paramecium is the primarily, its primary function is as a conduit for blood vessels, neurovascular bundles, blood vessels and nerves that are going to be traveling to the individual fascicles and innervating them and bringing them the nutrients and carrying away the metabolic wastes. And then within a single muscle fascicle, there are many muscle fibers. And these muscle fibers are individual cells. And an individual cell is surrounded by endomysium. So we had epimysium, paramecium, endomysium. Right? When we get to nerves, we're going to look at the anatomy of a nerve, and it's going to have epineurium, perineurium, and endoneurium in much the same way as these muscles have this mesium referring to connective tissue surrounding a muscle with the appropriate prefixes. So the endomysium is going to be the conduit for the capillaries that will be servicing the individual muscle cells and the axons of the terminal peripheral nerves where the motor end plate, where the neuromuscular junctions are going to form. This is also where we're going to find myosatellite cells. These are stem cells that help repair any damage to a muscle. Say you tear a muscle, you have a tear, a strain, or some sort of laceration. It's these myosatellite cells that will differentiate into mature muscle cells. And they live in the paramecium, or the endomysium, pardon me. All right, skeletal muscle fibers, the individual muscle cells, are enormous. They're enormous. Some of them can be up to many centimeters long, and they develop through the fusion of many myoblasts, or muscle stem cells. Now, and that's how they end up with being multinucleate. So here in panel A, up in the corner there, we have a bunch of myoblasts that begin to self-associate. Their cell walls fuse, they become syncytial, and all of these myoblasts are then now a muscle fiber with the nuclei spread throughout the length of these quite large cells. What is the rationale? Can you give me a biological rationale for that? why would it be an advantage to a cell to have many nuclei, or in particular these cells to have many nuclei versus a single nuclei? What do you think? Yeah.
I don't know if this is right.
It's okay.
If it were to tear, you would have something to regrow from.
Yeah, exactly. So these muscle cells are huge. They're very large, which is an advantage in terms of contractility, right? So you have a long span across which to contract. But because they're so large, if you have any kind of damage to the cell, or if you decide to take up bodybuilding or whatever, you need to put on muscle mass quickly. If there was only a single nuclei, it was down here, and you needed to repair some damage over in this part of the cell, you'd have to do all the transcription over here and the translation and then ship all those products to the far end of the cell. It's just logistically more convenient for the cell to be able to do its transcription anywhere in the cell that there's a call for. So the cell can grow quickly in a relative sense if there's a call for that, or repair damage quickly, which is also important. Does that make sense? All right. Yeah, so here's a number I put down. They can be as long as 30 centimeters long. That's pretty long. That's about that long. I mean, a single cell in your body is that long, maybe in your thigh or something like that. That's significant. That's a huge cell. All right, so let's begin to look at some of the structures in one of these remarkable cells. They're red because they are packed with myoglobin. Myoglobin is a monomeric relative of hemoglobin. We'll talk about hemoglobin, I think, in the respiration chapter, probably. I think that's the one I talk about it in. Myoglobin is essentially an oxygen storage molecule that it acts as sort of an oxygen buffer, right? If you suddenly need a quick burst of oxygen because you're running away from a werewolf and the lungs cannot supply that oxygen rapidly enough, there's this short time window of myoglobin, these reserves that can be called upon. They have multiple nuclei. We talked about the anatomy in lab yesterday. There's a sarcolemma, and it is a change in the transmembrane potential of the sarcolemma that leads to an excitation and eventually, which eventually becomes a contraction. The sarcoplasm is just the name we use for the cytosol of these cells. The sarcoplasm is loaded with glycogen. What is glycogen? Anybody know what glycogen is?
Storage molecule.
It is. It's a storage molecule. What does it store? Anyone else want to take a guess at that? Glucose. Yeah, it's a glucose storage molecule. It's a long chain of glucose molecules. Glucose is like I guess it's like the $20 bill of the body. If it's the $20 bill, then ATP is the dollar bill. All right. Glucose is broken down to make ATP. There's a ton of it stored. The two places in the body that store a lot of glycogen are muscle tissue and the liver. You store a lot of glycogen in the liver. All right. There are these transverse tubules, which I mentioned in class yesterday. The transverse tubules are basically just tunnels that push in from the surface of the cell. So the inside of a transverse tubule is the same surface as the outside of the cell. It's just a little hollow that goes into the cell. So these transverse tubules allow the entirety of the volume of a muscle cell to go through contraction simultaneously without having to have some sort of signaling cascade penetrate from the surface of the cell down into the interior. So really what it does is it increases the surface area to volume ratio of one of these cells, all right? The transverse tubules are right next to another vesicle delimited compartment, a membrane delimited compartment called the sarcoplasmic reticulum. It is the muscle version of the endoplasmic reticulum. It is a massive repository for calcium. It's where we store calcium in the cell, and calcium is going to become an important player in contraction. At the ends of them at the ends of these sleeves of sarcoplasmic reticulum are these terminal cisterna. A cistern is a place where you would store water, for example, and so there are these terminal storage balloons that are right next to, they're adjacent to the T-tubules, and because of that, they're called triads. The two terminal cisterna and a T-tubule are called triads. The sleeve of sarcoplasmic reticulum and rings of T-tubules surround a myofibril. These are bundles of protein filaments, the so-called myofilaments. Yeah, and the last thing is that there are tons of mitochondria. In here, in this diagram, they're indicated in purple. What is a mitochondria used for? Thank you. Beautiful. It's for ATP production. This is where all that glucose that's stored as glycogen is going to get broken down and converted. Actually, the glucose doesn't get broken down in the mitochondria, but the byproducts of central metabolism get broken down to make ATP. So we'll actually touch on that fun stuff a little bit later. Sarcomere structure. Sarcs from the Greek for flesh, meros parts, the flesh parts of the cell, the fleshy parts of the cell. This is a better diagram than I had in the lab. This is the next thing on my acquisition list for lab. I want to get a good model of this thing. So we have the thin filaments that represent the I band, the thick filaments of the A band, I meaning isotropic and A meaning anisotropic. So those words just refer to how they bend light. Something that's isotropic bends light exclusively in one direction. Something that's anisotropic bends light indiscriminate of direction in any direction. So things that have a handedness to them are going to be isotropic, and things that do not have a handedness, like a left or a right-handedness to them, are anisotropic. And we'll see when we see the molecular structure why this is the case. This was a designation given by microscopists before we actually understood the biochemistry of the molecular biochemistry, the molecular biology of a sarcomere. Running down the middle of the I band is the Z line, called a Z line for Zusamen. It's just a zigzag line, so that'll help you remember. It's a Z line. We'll see in the cartoon it really does look zigzaggy. Down the M is the M line. Down the middle is the M line, M for middle. Down the middle of this A band, there's this H band, this narrow region in the middle of an A band. This is where we have thick filaments without any thin filaments present. Okay, so this right here is the zone of overlap. This is all thin filament. This is thick filament. This is thick and thin filament. This is only thick filament. Does that make sense? All right. So here is a cartoon that might make all of that a little bit easier to understand. I don't know if any of you watched Tintin when you were little kids, but this is not Tintin, it's Titin. And it's a little protein that connects from the Z line to the end of the thick filaments and helps keep these thick and thin filaments in line with one another. It's the interaction of the thick and thin filaments that causes contraction. We're going to see that in a moment. So these are just some cross sections helping you to maybe a different way of visualizing the superstructure. This is down the Z line, and we see where Titin is attached to this network. We have the thin filaments here with the Titin in between. Here is the M line, which is only thick filaments. Here is somewhere in the H band. And this is the zone of overlap where the thin and thick filaments are able to interact with one another. All right, so let's talk about the microstructure of these thin filaments. A thin filament is actually called F-actin, filamentous actin. Actin is the monomeric protein that self-associates to form these long filaments that then the two filaments wrap around each other around this core of another protein called nebulin. So this little cable down the middle acts as the core around which two strands of F-actin spin. There is a handedness to this. All right, this is a left-hand helix, which is why it twists light in the way that it does. If it spun the other direction, it would direct light in the opposite way that it does. So that's why it's isotropic. Tropomyosin and troponin are two important regulatory proteins that sit on the surface of the F-actin, along the outside of these helical strands. So tropomyosin is this long, braided, double-stranded protein that is blocking an active site here depicted in yellow in the actin. It's blocking the active site from interacting from what will be the myosin, the thick filaments. All right, this is all thin filament. So the tropomyosin is blocking that interaction, and it's held in place by troponin. Troponin is a small globular protein, which is basically just stapling the tropomyosin down onto that helix. And troponin is itself regulated by the presence of calcium 2+. So when calcium is available, it's going to bind to troponin, roll tropomyosin off this active site, and then we can have contraction. So calcium is going to act as the molecular trigger on this whole show. The thick filaments are called myosin. So a myosin is a dimeric. So a myosin subunit is dimeric. We have two head groups that are next to each other with a hinge region and then a twisted tail. These are then bundled together like someone trying to stuff too many golf clubs into a golf bag. And at the end of this strand, we see the spring-like Titin, which helps keep everything aligned and helps with contractility or extension after the contraction is done. All right. So that's just a small look at the cast of characters. There's a whole host of other accessory proteins that associate these myofibrils with the sarcolemma, because there would be no effective contraction if these contractive bundles were just like contracting inside the cell but not actually squeezing the cell down, right? We need to like transmit that contraction to outside the cell. So it's the job of all of these these proteins here. There's a lot of them. This one in red here is called dystrophin. It's so called because a mutation in the gene for dystrophin leads to muscular dystrophy, as depicted in this individual here. So their muscles contract properly, but it's not that contraction in the cell is not able to be transmitted to the outside of the cell, where the whole muscle is going to be pulling on the connective tissue, which pulls on the bones, which allows actual contraction to happen. A lot of links in the chain to a healthy human being. So this is a little trip out I have. It's this idea that muscle tissue is fractal in structure. And so what is a fractal? Fractal means a scale invariant self-similarity. So that means that no matter how, like a truly perfectly fractal system, no matter how close you get, you see recurring patterns again and again as you go closer and closer, or further away or further away. And it is, muscle up to a point is certainly fractal. So we look at a skeletal muscle. A skeletal muscle is a big bundle of muscle fascicles. A muscle fascicle is, which is surrounded in epimysium. A muscle fascicle is a big bundle of muscle fibers, which is surrounded in paramysium. A muscle fiber is a big bundle of myofibrils, which is surrounded in endomysium. A myofibril is a big bundle of myofilaments, which is surrounded in a sleeve of sarcoplasmic reticulum. A myofilament itself is made up of, or a sarcomere is made up of these myofilaments of actin and myosin. So there is this sort of cascading pattern from the microscopic to the macroscopic in a muscle that I find kind of interesting. All right. So a muscle contraction, as I've already mentioned, is an interaction of a thick and thin filament. In particular, where these two filaments overlap. We see here a contracting muscle where the I band is in pink. It's very small. This is where there's only thin filaments. This is a contracted muscle. There's not much I band left because the zone of overlap has increased. The A band stays the same size, but we increase the zone of overlap and thus decrease the I band. All this stuff in green here is the glycogen vacuoles that are important for energy production. All right. So what's actually happening? The big picture is the sliding filament theory, that the filaments slide over one another. The thick filaments stay in place, and the thin filaments sort of work their way along, and the whole bundle contracts. The thin filaments slide towards the M line along these thick filaments. As I said, the width of the A band stays the same. The I band gets smaller as the zone of overlap increases in size. The Z lines come closer together. So here is it's me making a sort of lo-fi animation. You guys see how that works. I used to make the students come up front and play a game to illustrate this, but games are over. All right, an overview of the skeletal muscle contraction. There's really 4 steps in any contraction cycle. There's excitation, excitation-contraction coupling, then contraction, then relaxation. Okay, and here we have the neural control starting out this whole process of excitation. Then excitation happens at the surface. there's this process whereby that excitation is transmitted into the cell, contraction happens, and then afterwards the muscle needs to release. This is the power of excitable membranes. Okay. Yeah, so that's redundant. I just said all that stuff. This is the 4 phases of contraction and relaxation. All right. So just to go over a picture to remind yourselves of the anatomy before we go into depth with how this happens, we have the motor nerve sending out the telodendria, terminating in the synaptic knob. The presynaptic membrane is loaded up with these synaptic vesicles full of a neurotransmitter, which in this case, in this type of synapse, is acetylcholine. So these are cholinergic receptors. Then there's the synaptic cleft, where the presynaptic membrane is opposed by the postsynaptic membrane that is loaded with these cholinergic receptors that are embedded in the sarcolemma right there. And we see that there are these invaginations along the surface of the sarcolemma. This is to increase the surface area and really give it a whopping punch. Because remember when I said yesterday that the excitability, the strength of a synapse is dependent upon the amount of neurotransmitter released and the sensitivity of the postsynaptic membrane, how many receptors there are. So this is loading that postsynaptic membrane with receptors by increasing the surface area. All right. This is a motor end plate. This is what it actually looks like in histology, and that would be a synaptic bouton right there with the little granules would be the synaptic vesicles that would be full of the acetylcholine. All right, so that's what you're looking at in that picture. The neuromuscular junction. Step 1 is the arrival of an action potential down the axon, down the telodendria to the synaptic terminal. This sets in motion a degranulation event, which is the release of the acetylcholine here in the left-hand panel. This acetylcholine comes into the synaptic cleft and then binds to these chemically gated sodium channels, which are shown in purple. The membrane suddenly allows for a massive influx of sodium ions, which begins to depolarize that postsynaptic membrane. See how that's working there? So the depolarization is indicated in red. Here is a picture of one of those channels. Acetylcholine binds, the channel opens, ions begin to flood the cell, sodium enters, and the depolarization happens, driving the resting membrane potential from negative 90 millivolts, which happens to be the resting potential of this membrane. It's not negative 70 like it was in neurons. Muscle cells sit at negative 90. And then potassium exits, and the resting membrane potential returns to negative 90 millivolts. So, pardon me. Once the membrane has been depolarized, other voltage-regulated ion gates begin to open. So the trigger is chemical, but then it gets propagated through voltage-gated ion channels once we have the depolarization. And it begins to spread away from the site of the original synapse and moves out along the surface of the sarcolemma towards the T-tubules. All right. Meanwhile, the acetylcholine has diffused away from the cholinergic receptors. And here in this panel, the yellow crescents are acetylcholine esterase. I talked yesterday about the recycling of neurotransmitters using acetylcholine as the example that I wanted to use. It breaks the acetylcholine down into choline and acetate. The acetate remains in the cleft, and the choline is taken back up to be regenerated in the synaptic bouton. So here we see the excitation, the depolarization of the membrane traveling over the surface of the sarcolemma down into the T-tubule, approaching one of these triads. So in the T-tubules in purple with the excitation in green, and these blue tubules next to it, those are the terminal cisterna of the sarcoplasmic reticulum. There is a close interaction between those two membranes. In fact, they have a channel of proteins that link the two of them. Once the depolarization happens, it opens up calcium channels. It opens up voltage-gated calcium channels in the surface of the T-tubules, the sarcoplasmic reticulum, and that calcium floods out down its electrochemical gradient. So both its chemical and its electrical gradient are driving it into the cell. The cell is highly anionic or negatively charged, so the calcium wants to go in to get with that, and there's a much higher concentration of calcium in the sarcoplasmic reticulum than in the sarcoplasm. All right. So here's a diagram of that happening. So that whole this process, that process of excitation along the T-tubule causing the release of calcium from the sarcoplasmic reticulum. This is the excitation-contraction coupling. So this is how we're going to couple the contraction cycle, which is going to be described in a moment here, to the excitation, which I just described. This is the excitation-contraction coupling. Calcium floods out, binds to troponin, which then rolls the tropomyosin off of the active site of the G-actin in these F-actin strands. And then as soon as that happens, as soon as the site is open on the actin, then myosin can come in and with the heads of the golf clubs, form a cross bridge, form a transient link between the actin and the myosin. All right? Once that happens, we're ready to go. It's contraction time. All right, so here is the contraction cycle. This was excitation-contraction coupling. Here is the contraction cycle. Resting sarcomere. Calcium comes in, binds troponin, rolls the tropomyosin off the F-actin strands, and enables cross-bridge formation. So notice what's happened here before we move on. Notice what we have here in the myosin. We've already gotten that first contraction for free. We've gotten it for free. There is ADP and inorganic phosphate bound to the head of that myosin subgroup. The ATP that had bound there has already been hydrolyzed. The energy of that hydrolysis has already been used to cock the head back. All right, so the spring, the mousetrap spring, has already been loaded. That first cycle of contraction has already been paid for. All it needs is to form a cross bridge to trip the trap and let that head pull itself along the track of the actin. So cross bridge forms once the myosin can approach the actin. As soon as that transient cross bridge formation happens, that stimulates, that enables the hinge to contract, and this is when actual movement happens, that the spring on the neck of in the hinge region of the myosin flips, and releasing the energy of the original hydrolysis of ATP. In so doing, it causes a conformational change, of course, but that kicks off the ADP and the inorganic phosphate. Once the ADP and inorganic phosphate come off, the cross-bridge formation is not structurally favored, and the cross-bridge formation, it comes off off the actin. Once the tripped head is off the actin, now the active site for the ATP binding is open, and ATP can come on and bind to the head group again. A lot of pretty coordinated dominoes in a row, sophisticated and subtle string of molecular conformational shifts that all have a functional role. Once the ATP binds, there is ATP hydrolysis, where that high-energy phosphodiester bond that I talked about yesterday gets hydrolyzed. And when that bond gets hydrolyzed, it releases energy that is absorbed in the conformational shift of pushing the head group back again, a conformational shift. So the hydrolysis, I don't like it being so far away. I like to touch it to give myself a pointer. That conformational shift is stored in the hinge region, and it's ready for the next cycle of contraction. So as long as, I'll say this, as long as there's still calcium bound to the troponin and the tropomyosin is off the active group, this situation here is the same as this situation here, step 1. So we can go through many cycles of this now. As long as there's calcium in the sarcoplasmic reticulum, it's just going to keep ratcheting itself one step at a time along an actin chain. All right? So we go through many contraction cycles until the calcium gets pumped back into the sarcoplasmic reticulum, until it gets resequestered, and the contraction, then the tropomyosin will roll back onto the actin, and you can no longer form cross bridges any longer. Okay? So the contraction cycle, it's like this. You can think of it like that. So here is an uncontracted muscle, and then it contracts. You can think of it as squinching in from both sides or going a long way from one side, whichever the fixed end of the muscle is. All right. So how long does this last? How long does it last? Depends on these factors. So what's the duration of the neural stimulus? Meaning how long are we degranulating acetylcholine into the synaptic cleft? It's not an indefinite amount of time. How much free calcium is floating around in the sarcoplasm that's been released from the sarcoplasmic reticulum? And what is the availability of ATP in the cell? Your neurons may be telling your body, your muscles to move, because you've been running away from this werewolf for hours now. And you may have lots of calcium on hand because your body is at this point burning bones to get away. But you have run out of ATP. Why do we need the ATP? Because we need to cock that head back so it can keep moving, right? So there's this is burning ATP. If we run out of ATP, that can't happen. All right. So then relaxation. I already kind of covered this a little bit. Acetylcholine diffuses away from the acetylcholine receptor, the cholinergic receptor, gets broken down by acetylcholine esterase, which are then resorbed by the synaptic knob. When that happens, there's no more depolarization of the sarcolemma, and there are these constitutive calcium pumps in the sarcoplasmic reticulum. That constitutive means always on. Something that's constitutive means it's always happening. There are these constitutive calcium pumps in the sarcoplasmic reticulum that start winning the day and sequestering the calcium back into the sarcoplasmic reticulum. Calcium diffuses away from the troponin, tropomyosin, active sites are covered, and we are done. All right, so here, there's an important point here. This calcium sequestration, I told you that both the electrical and the chemical gradient are pointing in the same direction for calcium. Calcium wants to be in the sarcoplasm, not the sarcoplasmic reticulum. So putting it back in the sarcoplasmic reticulum is an active process. That constitutively active calcium pump is an ATPase. It's an ATPase. It's burning ATP. So you need ATP not only to contract a muscle, but also to relax a muscle. Okay. This is one of, so like muscle fatigue and muscle exhaustion, particularly in extreme athletes, is a pretty, it's a field that has gone through some real revolutions in the last few years, a new understanding of what's actually happening. People are like, oh, you're just building up lactic acid from central metabolism. That's actually not the case. Well, it is the case, but that's not the problem. But we don't have time for that today. All right, so then there's also elastic forces, just the elastic forces of a muscle opposing muscle contractions. I lift this, but then I use the opposing muscles in my triceps to put my arms back down. Gravity is always on your side. Okay, so, and this is just a recap of everything that I just talked about. So this is a nice summary table that you can look at. I'm not going to go back through it because I want to make sure to get through everything today. But yeah, this is just a recap of everything I've talked about. Rigor mortis. It's a fixed muscle contraction that happens after death. And this is due to a cessation of action in the ion pumps. There's no more ATP. All right? So what happens is, since there's no more ATP, we cannot continue to pump calcium back into the sarcoplasmic reticulum, and calcium begins to build up in the sarcoplasm, all right? And the calcium is going to bind to the troponin, it's going to roll the tropomyosin out of the way, and all the myosin is going to come in and bind and contract, but there's not going to be any more ATP for it to recock and continue contracting. So it's going to stay in this contracted position, but we cannot get the calcium out of there. The calcium is going to stay on the troponin because there's no more ATP to get rid of it, put it back in the sarcoplasmic reticulum. So we go into rigor mortis. It's a type of tetanus where the muscle is just heavily contracted. With time, that will release. That does release with time. Okay. Tension production. It's the all or none principle. Much like an action potential in the excitable membrane there's the all or none principle here. A single muscle fiber, meaning a single muscle cell, is either contracted or it's not. Part of the muscle cell isn't like, I'm going to contract and the other doesn't. No, the whole membrane is excitable. If any part of it goes, it's all going to go. It's a big stack of dominoes. So how do we get the partial contraction in a muscle like this, where it's not using its full capacity? Like I'm contracting this muscle, but it's not at its maximum, right? How do we get a variation in muscle tension across a muscle if a muscle fiber is either contracted or not, right? We're going to talk about that. And this is a part of the role of fascicles in the muscle superstructure. The tension, however, in a single muscle fiber, in a single cell, how much tension one cell is giving you depends on a number of factors. First of all, that's the number of pivoting cross bridges. How many cross bridges are pivoting? All right, and that is related to how much overlap there is. Is there a lot of overlap? Is there minimal overlap? That's going to be related to the amount of tension that you can build. What was related to that is what was the resting length? What was the length of that muscle before you started contraction? And we'll see why that's important in a bit. And then the final thing is how frequent is the nerve trying to stimulate that muscle fiber? This frequency of stimulation can have consequences in terms of the total tension that a muscle fiber can have. All right, so first a few definitions. Treppe, T-R-E-P-P-E, treppe, is a stair-step increase in the twitch tension. So a twitch is a single contraction cycle for a muscle fiber. A twitch is a single muscle contraction cycle. All right, stimulation, contraction, 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 acetylcholine goes away, relaxation. That's a twitch in one muscle fiber. So if you look at the tension in that single muscle fiber, if you have multiple contractions, you can step the overall tension that muscle fiber is giving up to a limit. All right. So this is due to repeated stimulations immediately after the relaxation phase. So it causes a series of contractions with very minimally. It looks like it's going up, but it's actually not that much. Very minimally increasing tension. All right. And this is due to variations in optimizations in the zone of overlap, like how much rubber is meeting the road in that muscle fiber. You're sort of conditioning it with multiple repeated rapid contractions. Then we can get, if the stimulations are so rapid that the beginning of one contraction cycle overlaps with the relaxation phase of another, we can get this wave summation. So each of these waves of contraction begin to overlap, and we step that tension up slowly. All right? It's stimulation before the end of the relaxation phase. If we just keep pushing this concept, right? If you keep doing that, you're going to get to a maximum level of, I shouldn't use the word maximum, but you're going to get to a plateau of contraction that is dictated by the frequency of the contraction cycle. However, if you are sending a nerve impulse, there is genuinely a werewolf after you, and you are contracting that muscle as fast as you possibly can, you're going to get to what's called tetanus. That is the maximum possible tension that muscle can give you. The nerve is firing as fast as it can fire, so you are in the relative refractory period of that nerve, and you're firing that nerve, and that muscle fiber is going through contraction cycles as rapidly as it possibly can. You'll reach this point complete tetanus, where the muscle fiber is under maximal tension that's possible for that muscle fiber. Not very sustainable. All right, so are there any athletes in the class? Is there anyone on any sports teams in here? A couple of you? Good. Well, this is a good thing to know here. Particularly as a yoga instructor, I like talking about this. This is the length-tension relationship. The length-tension relationship says that there is an ideal length in a muscle that is going to give you the maximum possible tension for the same muscle mass. All right? And this is wholly dependent upon having optimized this zone of overlap. All right? You may think that over here in section C, this would be ideal because you have a lot of overlap. You have pretty much the most overlap possible between the thick filaments and the thin filaments. There's a lot of little head groups on the ground ready to contract. But there's nowhere for them to go. There's nowhere there for them to go. So, and then over here, when a muscle is super stretched out, like you've torn a muscle, there is not a lot of overlap. There's not much rubber on the road to actually generate much tension. You can go anywhere you want, but there's not a lot of actual chemical interactions, physical interactions to create the force. So there's this sweet spot in here where you have the optimal overlap with lots of room to yet contract into. Okay, so this is to say that for an athlete, without adding any additional muscle mass, you can get higher performance out of your muscles. You can get higher yield, more strength out of your muscles just by stretching them properly, getting them out to the right resting length. When you're going into an active session, if you can warm up, then stretch, and then have the activity, you'll get the most out of the activity because those muscles are now at the proper length. A lot of times you see these bodybuilders, not bodybuilders, weightlifters, that are super muscle bound. They've got all of this muscle on them, but actually they may not be the ones that are lifting the biggest weights in the Olympics or whatever. A lot of times you go and see the guys that are lifting the biggest weights at the Olympics. It's these very lithe guys that seem pretty fast on their feet and pretty loose. They're getting the most out of their muscle mass because they have mastered this length-tension relationship. So that's an important thing to think about. Yoga can, in fact, make you a better athlete. All right. So now, moving from a single muscle cell onto a whole muscle, how do we get a variable amount of tension in a whole muscle? Well, there's this notion of a motor unit. A motor unit is an ensemble of multiple muscle fibers that are under the control of a single neuron. Any muscle fiber under the control of the same neuron is all going to contract together. But a muscle fascicle that is many muscle fibers is going to have multiple nerves, multiple motor units servicing it. And the way that you get a variable amount of tension in a muscle is by the selective recruitment of different motor units. Are you using one motor unit, or 2, 3, 300, 5,000, 10,000? How does that work? And this is another way that the tension of the different motor units add up. So here we go with three different color-coded motor units. One of them contracts, motor unit 1 contracts, and then releases. Meanwhile, motor unit 2 has begun to contract, and is adding the additional tension that it's providing to the whole muscle to the ensemble. The dark red line is the overall tension of the entire muscle. So it's a sum of the action of all the different motor units. If you need higher tension in a muscle, you just add more motor units, or you increase the rapidity with which a motor unit is being engaged. Does that make sense? Should be pretty straightforward. Phases of a twitch. Yeah. Well, I probably should have had this one before the last slide. It's just we have the resting phase, stimulus, the contraction phase where we're increasing tension, the relaxation phase where we're burning ATP to put the calcium away, and the reduction in tension. We'll leave those panels. That stuff's pretty redundant there. Okay, so the time course of any of this tension production can depend upon the type of muscle fiber. There are fast, intermediate, and slow muscle fibers. You may think to yourself, well, why are they all just fast? Well, slow muscle fibers have advantages in terms of their ability to be aerobic. A slow muscle fiber has different metabolic, its metabolism is tuned a little bit differently so that it can remain in the aerobic region much longer than a fast muscle fiber can. Okay? So, for example, an eye muscle is a very fast muscle fiber, whereas the gastrocnemius is intermediate, and the soleus, that deep, big muscle of the calf that's responsible for helping you run from a week's worth of werewolves, is a very slow muscle fiber. All right. Muscle tone. The normal tension and firmness of a muscle at rest. Muscle units do maintain body position without motion. And the more your basal muscle tone increases, metabolic energy is used even at rest. However, this guy who'd like you to think that he's at rest and is ripped at all times is probably not exhibiting basal muscle tone, right? So your muscles, they're not completely flaccid at rest. They do have some muscle tone, but most people aren't fully ripping it all the time. That guy. Okay, the types of contraction. There's two types of contraction, and this is just totally physics, macroscopic contraction mechanics. There's isotonic contraction and isometric contraction. So an isotonic contraction is a contraction where the tension is the same, the muscle tone is the same throughout the contraction. So my bag is not changing in weight, and I'm going to lift it up just like this. Okay? So this is isotonic. The length of the muscle is changing. The length of the muscle is changing here, but the tension is not. The tension is always the same in that muscle. That's isotonic. Isometric contraction is one where a muscle is contracting, but as it contracts, it's increasing the amount of tension in the muscle, but the muscle is not changing in length. This happens when you are lifting something that is resisting the force of the tension building up in the muscle. The resistance is greater than the applied force. All right? When the resistance is greater than the applied force, it's called isometric because the length of the muscle doesn't change even though the tension is going up. You're not actually able to move the thing that you're trying to move. But isotonic, the tension is greater than the resistance, and so the length in the muscle is able to change. You're able to move whatever you're trying to move. So most contractions, all contractions, have isometric and isotonic components to them. So when I'm going to lift this bag, there is a brief period of time where I have not, my muscle has increased tension in it, but the tension is not greater than the resistance. Right now I'm in isometric contraction until that tension gets to a point where it exceeds the resistance, and now I've passed into an isotonic contraction, where the force is equal to the resistance, and the length of the muscle is getting shorter. Does that make sense? It's not really that important, but all right. So the resistance is inversely proportional to the speed of contraction. That should be pretty self-explanatory. I don't want to spend too much time on that. Let's get into the business here. All right, how do we pay for all this? How do we pay for all this? I'm a biochemist, so we're going to talk about some biochemistry. Basically, we're all little bonfires running around. So over here, we have a carbon source, the logs, sucking up oxygen from the atmosphere, combusting the oxygen, and releasing carbon dioxide and water and energy in the process, okay, which is the fire, the heat. That is exactly what's happening here. That's what we're doing. Instead of logs, we eat power bars, and the smoke that we give off is our breath. The energy that we give off, our breath, and the water is absorbed into the fluid, the aqueous environment of our body. It's not directly the stuff that we breathe out, but and then the energy is what we use. The fire is what we use to make motion happen. All right. So yep, there's that. Little bonfires running around. So there's aerobic metabolism and anaerobic metabolism. I'm going to talk about the basics of the energy economy associated with this. Has anybody in this class had this stuff before? Maybe one. Okay, so this is new for everybody. Great. Here's a muscle cell. There's a blood vessel on the top. We've had our delicious whole grain breakfast. There's lots of glucose in our blood. We're breathing. There's oxygen, but you're sitting there. You're not running from werewolves or lab exams. So the glucose gets imported into the muscle cell with the glucose transporter due to the presence of insulin in the blood. And glucose, when you are resting, your body doesn't actually need that much glucose. So we're going to put it in the bank. We're going to bank it. Some of that glucose is going to be stored as glycogen. It does require a little bit of ATP to produce the glycogen. Another thing we're going to be doing is storing some of the energy of ATP as creatine phosphate. Has anyone heard of creatine before? Like all these bodybuilders take creatine. Creatine is an energy storage molecule. We can take the phosphate off of an ATP and store it on a creatine, and then recycle the ADP and put another phosphate on it. So it's a quick, readily accessible store of high energy. All right, and it doesn't really do any other thing besides store the energy. ATP is doing all kinds of stuff. It's like a valuable currency. The creatine phosphate is sort of like a mutual fund that you can tap when you need it. All right, and where are we getting this ATP? We're getting this ATP from the fats in our blood. The fats are going down into the mitochondria, going through beta oxidation. We may get a chance to talk about that today, probably not, probably run out of time. But The fats are getting chopped up one carbon at a time and producing CO2. And the energy from burning those fats is being used to recycle the ATP in the mitochondria to help store the glucose as glycogen and some of the phosphate energy as creatine phosphate. Capiche? So there's the carbon is circled in is the G in green, the oxygen is next to it, and then the output is the carbon dioxide. So just so that you can, I don't know, maybe it's not amazing to all of you guys, but the carbon dioxide that you breathe out, maybe not all of you have appreciated this, the carbon dioxide that you're breathing out is not carbon dioxide you've breathed in. It's carbon dioxide that you are burning off. It's you ate that carbon. That was your breakfast. You are breathing your breakfast out. Okay, you've burnt it. You've combusted it with oxygen, and you're breathing it out as carbon dioxide. Of course, there's carbon dioxide you've breathed in that you're breathing out with the carbon dioxide from metabolic sources, but the extra carbon dioxide that's coming out is from your cornflakes. So this is the creatine system in the so-called phosphagen system. Phosphagen meaning phosphate generating, high energy phosphate generating system. Here's creatine. There's this enzyme called creatine phosphokinase. A kinase is an enzyme that takes a phosphate group and sticks it on another. It takes a phosphate group and sticks it on something else. All right. So a phosphatase is an enzyme that does the opposite. It takes a phosphate group off of something, but a kinase takes some compound and sticks a phosphate group on it. Here, it takes the phosphate from ATP, making ADP, and creates creatine phosphate. So this is CPK, creatine phosphokinase. And then when we need it, where am I at? Jeez. When we need it, we can get that high energy phosphate back very quickly. It's a very quick source for phosphate energy. There's also myokinase, which is another kinase that's responsible for quickly taking a phosphate off of ADP and tacking it onto an ATP. So myokinase takes two ADPs, which aren't in themselves very useful, and makes one an AMP and the other an ATP. All right? So it says, look, there's two of you guys with half of what we all need. We're going to take everything and put them in one person. The other guy's going to figure it out elsewhere. All right. So now we're going to get out of our chairs and we're going to actually do something and burn a little bit of energy aerobically. All right. We're getting on the treadmill. Go nice and slow. So at this point, we're using oxygen. We're liberating the glucose that's tied up in the glycogen in our muscle cells. That glucose is going to go through what's called glycolysis. Glycolysis, the breaking down of glucose. That process of glycolysis releases some ATP. It converts ADP to ATP. We're not going to go into the details of that. And the breakdown product, so glucose is a 3-carbon sugar, or 6-carbon sugar, and pyruvate is a 3-carbon fragment. So you get 2 pyruvates out of 1 glucose. Pyruvate is then able to be taken up by the mitochondria, and pyruvate feeds into the citric acid cycle, the Krebs cycle. This maybe some of you had that in biology in high school. It's this cycle that helps produce NADPH that goes into the electron transport chain. Basically, the upshot is there's a lot of, this is a very highly simplified cartoon. There's a lot of chemical steps in all this, but the upshot is for one glucose, we're getting 36 ATP altogether. So 2 of them in glycolysis and 34 of them out of the TCA cycle, the carboxylic acid cycle, and the electron transport chain. Right. That ATP can then go on and do what it needs to do in terms of contraction. All right. And to make this happen, however, we need oxygen to be able to come in. All right. Oxygen has got to come into the mitochondria. 2 of the ATP don't require oxygen. 34 of them do. All right. And then the output is carbon dioxide. We're burning carbon from glucose, and we're still burning carbon from fat. All right. So now it's the end of the race. You know, we're doing the 5K. We've got another 200 meters on the race, and we're going to kick it in. And we start really running. Anaerobic activity. Peak metabolism, you're running as fast as you can make your body move. All right. At this point, your cardiovascular system is not able to get oxygen to the cells fast enough. You just cannot move the volume of oxygen that you need to keep it aerobic. So what happens is the mitochondria goes out. I mean, it's still doing its thing, but it's not meeting the demand. So the bulk of what happens, the bulk of that pyruvic acid, instead of going into the mitochondria, just becomes lactic acid. It becomes lactic acid and gets exported out of the cell into the blood and goes to the liver and gets recycled. At this point, we start dipping into the phosphagen system. So all of that phosphate that we were storing up for a rainy day in the creatine starts getting pulled out, and that CPK reaction, the creatine phosphokinase reaction, starts going backwards. We take ADP, which there's a lot of being released from the myosin head groups and the muscle contraction. We take that ADP and we're going to rephosphorylate it, generating the ATP. This is why bodybuilders like to take creatine supplements, because they're not really doing aerobic activity. They're doing anaerobic activity. They want to load their cells with creatine more than is metabolically normal, and they load their bodies with creatine so that they can maximize their anaerobic power, because they're not lifting for hours on end. They've got 30 seconds for an intense clean and jerk, and that's all they need the energy for. All right. So here's a table that talks about some of the specifics of that. I'm not going to go through all the numbers. Okay. Here's kind of the same thing, but in a cartoon form. In the beginning, you're starting out aerobic respiration. You're using oxygen, but you're speeding up. You start to liberate some of the oxygen out of the myoglobin that's being stored in there, so we're able to dip into that extra oxygen that's higher than the amount that our cardiovascular system can bring in. Then we have the phosphagen system kicking in here, the creatine and all that. And then we get beyond our capacity to deliver oxygen, and we sort of short circuit the mitochondria and begin to build up lactic acid. Finally, we're done with our sprint. At the end, when we stop, we're not moving any longer, but we're still like, we're paying back the oxygen debt. All right, we stripped all that myoglobin off, the oxygen off the myoglobin. Our body wants to put it back. we're trying to catch up with the metabolic debt that we were paying. Okay, so here's a picture of that, a nice chart. God, these bulbs are really crummy. So this supposedly pink area here where it says oxygen deficit, during the beginning of the sprint, we're not transporting enough oxygen, right? And we reach a steady state, we stop, we have to pay back that oxygen deficit that we burned, right, like off the myoglobin or whatever. And so that's what happens at the end of the strenuous activity. So the green area should equal that red area. The recovery period, Cori cycle. Let's not go into the Cori cycle. It'll take a long time to actually get into your heads the right way. Yeah. So the point here is simply that the amount of power that you can generate at any one given time is inversely correlated to your endurance. All right. So you can have really high power, like a weightlifter can have really high power, but they cannot do that for a very long period of time. Whereas a marathon runner actually is not using a huge amount of energy within a short period of time. But it's their endurance. They have a very long endurance for that. What was it? Muscle fibers. Yeah, right. So the factors, I should say this is interesting. So the factors are the types of muscle fibers involved. Are we talking about fast, intermediate, or slow muscle fibers? And then your personal physical conditioning is also going to affect the power-endurance relationship. All right, so muscle fatigue. So when your muscles are fatigued, there's a decrease in ATP synthesis. This can be because you burned up all the glycogen in your cells, which is actually good for you. It's good to clean out the metabolic closets every so often. You've damaged the sarcolemma and the sarcoplasmic reticulum. In particular, it's these calcium channels. There's been research that shows that this kind of thing that this guy's feeling here is not due to a buildup of lactic acid. Instead, it's because there's been damage to the calcium transporters in the sarcoplasmic reticulum, which leads to leaky sarcoplasmic reticulum, and that the muscles cannot relax properly any longer, nor can they contract properly any longer at a certain point. It takes time to repair the damage. This is certainly exacerbated by a dropping pH due to an accumulation of lactic acid, which we saw was a byproduct of pyruvate production. This is going to mess with the functioning of a bunch of enzymes. And if you have a lot of action potentials where you're doing wave summation, where the excitation phase is overlapping with the relaxation phase of a previous twitch, then you never let the sodium-potassium ATPase get that extracellular electrochemical gradient back to where it needs to be. And what happens, you end up shuttling too much potassium out of the cell. You pump all the potassium out of your cells, which is why they say if you're going to be like you're having an athletic thing, you're having a track meet or whatever, to load on some potassium that day to make sure you have plenty of potassium on hand, because repeated action potentials tend to export all of your potassium. You have the potassium, but it's not in the cells where it should be, so it can be released. Yeah, and then you use up all the acetylcholine. The nerve fibers are sort of taxed beyond their ability to recycle the neurotransmitter. It starts to hurt. Okay. So high-intensity exercise, men and women are... So before I get to that part, it's determined, your ability for high-intensity exercise is determined by this intrinsic factor called your VO2 max. We are going to measure our VO2 maxes in the class. I designed this metabolic lab that we'll do in a couple weeks, I think Monday, a week from Monday, or Tuesday, one of those days. So that is the amount of, the VO2 max is the maximum amount of oxygen that your cardiovascular system can transport to your muscles within a given period of time. Right? So the higher your VO2 max, the greater your high intensity capacity is. All right? So like really peak athletes, really top tier athletes have exceptionally high VO2 maxes. And you guys as young people probably have much higher VO2 maxes than you will have when you are in your 50s, for example. VO2 max does go down a little bit with time. Tends to be greater in males than females, but as I say there, it can be twice as great in a trained endurance athlete, even more. There are some people out there with these VO2 maxes that are just phenomenal. So let's make enough of that. Yeah, so the difference between these fast twitch and these slow twitch muscles is in the ratio of the different fiber types, the fast or the slow fiber types that are found within a certain fascicle in that muscle. All right, so it's like it in fact is the difference between white meat and dark meat in a chicken. White meat would be like the fast twitch, and the dark meat would be like the slow twitch. So the slow muscles are ones that have more endurance, and the white meat would be one that is more anaerobically trained. And that makes sense in a chicken. They've kind of lost the ability to fly. The chicken breast is not actually built for endurance the way the breast of, like, say, like a goose, like a Canadian goose. They're essentially all dark meat, and they can fly long distances. It's kind of the same here in humans. There's no white meat on humans per se, because there's plenty of myoglobin in all of our muscle cells, but there is a difference. There is a difference in the ratio of fast and slow muscle fibers. And there are also genetic differences between people. Certain people have more of one type of muscle fiber than another. It can be ethnically correlated. Certainly, people who have a particular ability to perform really well, like, for example, Kenyans, that culture, that ethnicity, as part of the genetic selection over the eons in the environment that they lived in, enables them to have a real endurance, and that is borne out when you look at the muscle fiber distribution in the muscle tissue. So these fast glycolytic, fast twitch, or type 2 fibers, they're adapted for quick responses. They are not fatigue resistant, where these slow oxidative muscle fibers, or type 1 fibers, are very fatigue resistant. They are not tuned for anaerobic response. All right. So resistance training, weightlifting, endurance training. So yeah, this is just the different types of training. You can encourage or tune one muscle fiber type versus another, whether you're having resistance training, working anaerobically with increasing the capacity of the fast response muscles versus the ones that have more myoglobin, the endurance training of aerobic exercise. This tends to encourage the slow twitch fibers. You have an increase in mitochondria accumulation in these cells, and it's going to increase your red blood cell count, the oxygen transport capacity, and thus the VO2 max of your body is related to your RBC count and your oxygen transport capacity. Yeah, okay. Let's see. So this is just a table sort of comparing some of the properties of slow, intermediate, and fast muscle fiber types. I'm not going to go through that. Oh, I'm almost done. So this is the last slide. So the pathology, amyotrophic lateral sclerosis. Yeah, this is a disease where the muscle fibers of a person's body begin to wither and die because of de-innervation. It's actually, I'm putting this in the muscle chapter, it's actually a nerve disorder. The muscles wither and die and waste away, but it's due to a de-innervation by the sclerosis of the axonal sclerosis. So they have recently found that, I guess not that recently, it was 2008, that was 7 years ago now, that creatine supplements can actually significantly increase life expectancy in people with ALS. That right there is my father. He died of ALS 4 years ago now. But it is, it's a shockingly common and pernicious disease. It's actually more common than a lot of people realize, although I guess in the last year, it's been in the last year that the ice bucket challenge was for people with ALS. So that was cool to see. Yeah, I guess that's all I have to say.